We know how to do in vitro fertilization for people. So we can take an egg and a sperm and put them together and then implant them into the woman. And so we've first tried this in animals, and then for some years now we've been able to understand enough about the process to be able to make it work in people. So to do this, though, you have an opportunity to study that egg and that sperm before you actually implant it. So after they're joined in the lab, then you can study those. What's most commonly done is if you're at risk for inheriting a disease. So if you know that this disease is transmitted through your family, you can have that embryo checked for the presence of that disease before it's implanted. And that's done by taking a tiny piece of the embryo and testing it by DNA testing, the same way that you see on CSI and those other shows where they amplify a piece of the very, very, very small fragment, a very small amount of it, and amplify it so that you have enough to actually study, and then sequence the DNA and check to see if the inherited change that causes the defect is there. So then you can select for those in which there is no inherited defect present. There's also certain things that are transmitted only through males or only through females. If you had a disease like that, you might do something a bit simpler and select for sperm and egg together that produced a particular sex, okay, whichever one was unlikely to inherit the mutation. It's kind of surprising that this works, because you would think the embryo would need every bit of the genetic material, but part of the embryo goes to make the nurturing tissue around the developing conceptus, and so you don't actually need every single cell that's present in that embryo to make a normal child. And so we can abstract a very tiny piece and use that for the testing.